Hi everyone, I'm delighted to be here today and to have the opportunity to present at London Calling. And today I'd like to share how we use nanopore direct RNA sequencing to address two challenges in pathogen genomics. So the first one will be on arbovirus surveillance, and the second one will be on uncovering a new antibiotic resistance mechanism in bacteria. So let's start with arboviruses. So arboviruses are RNA viruses, such as dengue and chikungunya, that are transmitted by mosquitoes. In the Pacific region, they pose an ongoing epidemic risk, and the current detection methods rely mostly on cDNA and PCR, which are limited by reverse transcription and PCR bias. So in collaboration with the Pasteur Institute in New Caledonia, we use direct RNA sequencing on viral RNA extracts of two viruses, so dengue and chikungunya, and we obtained full-length genomes in single reads up to 10 kb, and we could also detect variants at a genome resolution. In addition, we tested different gradients of inputs for our library preparation, ranging from 97 nanograms down to undetectable levels, and we still were able to obtain sequencing data. So this is also very promising to analyze low viral load samples, and is also highlighting that low input doesn't necessarily mean no output. Now, onto the second project. It actually began 15 years ago when two collaborators studied a bacterial strain that was resistant to erythromycin, but was lacking any known resistant gene. So back then, comparative genomics identified a novel gene, potentially encoding for ribosomal RNA methyltransferase. But back then, traditional approaches failed to characterize the gene's mechanism of action, and the project stalled. So fast forward to today, now, with nanopore sequencing, we have the ability to sequence native RNA and detect modifications. So we decided to bring this project back to life. We cloned the resistant gene into E. coli, and we used nanopore direct RNA sequencing on two strains, one with the gene and one without the gene. We then used Dorado and Modkit to do the analysis, and we looked at the percentage of modifications we could detect on the ribosomal RNA. What we found is that we see an elevation in the M6A signal in the strain expressing the gene compared to the one not expressing the gene. So this result suggests there is a methylation-based resistance mechanism that is potentially disrupting the antibiotic binding on the ribosomes. So in summary, during this talk, I presented two applications of nanopore direct RNA sequencing. The first one in virology, where it enabled full-length genome resolution, enabling real-time surveillance and viral evolution tracking. As next step, we'd like to test if we can use this method for other arbovirus surveillance, and maybe also integrate it into a One Health approach. And the second application enabled us to sequence native RNA and access modification, uncovering mechanisms that we couldn't detect before. As next step, we'd like to investigate the precise modified positions on the ribosome and also validate the gene function by deleting it in the wild-type strain. And so ultimately, direct RNA sequencing is a very powerful tool that is reshaping how we do pathogen genomics. With this, I would like to thank everyone involved in this work, and thank you all for your attention, and I'll be happy to answer any question.